Alright, guys and girls. So, today we're going to do a little flipped classroom stuff. Now, what that really consists of is me doing some notes, just like I would in the room, except this time we don't do them at the house. Save us a little time in class, that way we can cover a little bit more material and do a few more things. Alright, so we've talked about cells, especially in 7th grade and before that. So this should be all pretty much review. Okay, so there are 3 parts to the cell theory. First, all living things are made of cells. Cells are the basic unit of structure and function. Now, we've talked about things like carbon, nitrogen, and hydrogen as elements and things of this nature, but they don't have a function as in working together and providing like a living thing. They help us live. We need carbon, we need nitrogen, we need hydrogen, but they're not the basic unit of something that is alive. Okay? And then third is all cells come from pre-existing cells. So, who are the guys that helped us figure this out? All right, first one, Anton von Leeuwenhoek. He was a businessman more so than a scientist, but he was really the first person to use a simple microscope. Okay, he looked at pond water and saw that there were little things in the pond water. These little things were like little amoebas, parameciums, little things like that. Now, Hooke was the first one to use a light microscope where the light source. So you got your, let's see if draw a quick little microscope here. Okay, so you look through the top part, see what's on the slide. So a light microscope, we shoot the light up from underneath, and he saw a slice of a cork, just like you would have in a wine bottle or anything like that. Now, they weren't called cells up till this point because what he saw were plant cells with cell walls and all that kind of good stuff. So what he had here was something looked like this, and so they look like the cells in a monk or a monastery. So that's why he called them cells, and that's where we get the term cells from. All right, last 3 guys. Schleiden, he was one that said all plants are made of cells, and of course we know that to be true. Schwann was the one that said, okay, cool, plants are all made of cells. Let's look at animals. Sure enough, all animals are made of cells. And Virchow was the guy that said and really saw cells starting to divide, and it's like, ooh, all cells come from pre-existing cells. All right, so some cell structure basics. All right, cell membrane is that phospholipid bilayer, and we're going to get more into the phospholipid bilayer when we talk about transport, but it is a flexible membrane that has a whole bunch of stuff in it. So we got our phospholipids, we got proteins, we got all these things that are going to help our cell maintain homeostasis, move things in and out. Nucleus, all right, we always, think about big nucleus. It controls, it has our DNA in it, all right, and we'll send out RNA too sometimes. And then we got the cytoplasm. Cytoplasm is all that material like in and around that area. So something down here in the south we have the classic fruit jello, right? We take a can of jello out of the or the fruit out of the can, dump it into the jello, and you end up with all these little pieces of jello spotted throughout, or the little pieces of fruit spotted throughout the jello. Well, the jello is kind of like the cytosol. It's the stuff that holds everything together. All right, we have 2 main types of cells. We have prokaryotes and eukaryotes. Now, with a prokaryote, big key thing here is no nucleus. Okay, and they're very, very simple cells, and they are small, very small. They're about the size of one of these organelles that we'll talk about next. So a prokaryote mainly we're just talking about bacteria when we talk about prokaryotic cells. Eukaryotes, more or less everything else. They contain a nuclei. They have cell membrane. They have cytoplasm. They have all this other stuff. They have a bunch of specialized organelles. All right. Prokaryotes really just have a little DNA in there, a little piece of DNA called plasmid. All right. Boom, 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 boom. All right. In there, circular. Like I said, it's kind of circular. That's why I drew it like that. And some ribosomes and some things of that nature, but not a lot of organelles. Okay. These are much more complex, larger. Of course, larger is a relative term because they're still microscopic. All right. So, kind of to give you a proportion of the size, the prokaryote cell here is about the size of a mitochondria. That's about it. It's smaller than the nucleus. So, it's very, very small comparative to a eukaryotic cell. All right, so some similarities and differences. Differences, we have a nucleus complex and have organelles. No nucleus, super simple. Both have cytoplasm or the cytosol and a cell membrane of some kind. Some have, and some actually have cell walls as well. All right, jump into it. Cell wall, only in plants. All right. Bacteria, some bacteria have it, algae, and fungi. So more or less, I should put maybe not only in plants, but maybe only in non-animal cells. So these plants are consistent, these cell walls are consistent of cellulose. They provide rigidity and structure to them. All right. We as omnivores can't really process cellulose well. Most herbivores can. So, that's why they tell you to eat lettuce and things, because it's fiber. This is kind of what we call when we think of one of the forms of fiber. So, as I said previously, animal cells do not have cell walls. Cytoskeleton. This is something that will help maintain the shape and structure of the cell. So microfilaments as a kind of sound or long, thin strands. So that would be these bad boys here. Okay. The microtubules, just like it sounds, is a tube-shaped structure. It's going to be right there. The control center, aka the nucleus. All right. So the nucleus houses the DNA and in the form of chromatin and chromosomes, and we'll talk more about that when we get to mitosis. And it also holds the nucleolus. All right. So in a nucleus, you will have, so the nucleus is the whole thing. Nucleolus is this smaller little portion here. This is where we're going to make ribosomes, which are going to do a crap ton of things in our body. Centrioles, these bad boys are great for, mitosis and meiosis. M&M. They play a role in cell division. They are only found in animal cells. Ribosomes, these bad boys make proteins, all right? Those important things we need in our bodies. We talked about them. We talked about with all the macromolecules, okay? They're made up of rRNA or ribosomal RNA. They're little pieces like this. We got one top, we got a bottom, and a top, and it's going to come along and help make all these proteins. Now, they can be free or just kind of floating around, like if this is our cell here. You just got all these little dots. Okay. Looks like cell has chicken box. Okay. Those are just free-floating ribosomes. They can also be attached to things like the rough endoplasmic reticulum. So, we got two types of ERR, endoplasmic reticulum. The first type would be rough, all right? This is with a whole bunch of ribosomes. and it exports proteins from the cell and sends them all out to wherever they need to go. Whatever kind of protein is made gets to go to where it needs to go to. The smoothie R, this has no ribosomes and really does more with lipid production and breaking down of toxic substances. So this one you can think of, it's very rough, or protein is a little bit more porous, a little bit more coarse, especially after you cook it. Oil or fats or lipids. All right, these bad boys are very smooth usually. I've never met a rough lipid in my entire life. So, all right, Golgi apparatus. This is kind of like FedEx, for lack of a better way to put it. It processes and modifies and ships everything all over wherever it needs to go. Okay, it does proteins, it does lipids, it has vesicles. Things will come in, things will come out. Like one side here, things will come into. It'll get processed as it moves through here, and then it comes out. And these bad boys right here, which are called vesicles. Lysosomes, these guys are designed to break things apart, break things down. So, you can have cells that just need to be like, though sometimes you see right here, we have like food particle coming in, getting broken down, starting to into digestion, starting to send to places and packaged to be go to wherever it needs to go to. You can also have little viruses come in. All right. They'll come into the cell and they'll get by the defenses, and they will, the lysosomes will come in and take those apart too. Also, if you have like an old mitochondria that's just not working like it should anymore, all right, it'll come in and it'll actually gobble this bad boy up, break it down into its usable parts, and kind of recycle it for later use. Well, not so much to reuse it as a mitochondria, but it'll take all its parts, the carbon, hydrogen, oxygen, the things that make it up, repurpose those into something new. Big old vacuole. Okay, this bad boy is only in plants and is the largest organelle for plants. As long as we're watering the plant well, it's the largest one. It kind of fills up with fluid, water, salt, sugars, things like that the plant needs to live, and it pushes out on the cell, causing turgor pressure. So if your leaves are looking droopy in your little house plant at the house, all right, it's probably due to lack of water, and the vacuole has shrunk from when it's nice and standing up all pretty. All right, chloroplast. Most people know about this bad boy. Oops, there we go. All right, chloroplast. He turns light energy or photons into chemical energy, which would be C6H12O6, which is sugar, right? So we do this in a process called photosynthesis. We'll go more into depth later on this one. And it has a pigment called chlorophyll. Mitochondria, the powerhouse of the cell. It's one thing everybody always seems to remember. Now, the key, what I want you to understand, and we all heard, oh, it's the powerhouse of the cell, blah, 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 blah. Well, what does that mean? Okay, so what this bad boy is going to do is going to take in food that we eat, and it's going to use that to send out ATP, or adenosine triphosphate. Now, adenosine triphosphate is very important because literally every time your muscles do this, okay, every time they move, use up thousands and billions of ATP. Okay, so yes, it is the powerhouse of the cell, but what it makes and what it uses and what we kind of get from it is this ATP. Now, both plants and animals have this because both plants and animals need ATP. Chloroplast, of course, and the last one is only plants, but both animals and plants need energy, so to grow, develop, change, right? And this is what we get out of this. So last little few random ones. We got cilia and flagella. So cilia are like little hair-like ones, kind of like this. They kind of move like oars back and forth. Flagella, on the other hand, are kind of like a circular thing, and they spin like a boat propeller in a circle, and they power it forward. So, some key differences between plants and animal cells. Once again, cell wall, chloroplast, large vacuole, no centrioles. So, this bad boy has centrioles, no cell wall, no chloroplast, no large vacuole. They have vesicles in here, like from the Golgi apparatus, but no large vacuole. See, this is our vacuole here. We got nothing like that over here. All right, we're going to end it right there. And if you have any questions, bring them into class and we'll be happy to answer.